what we're going to do is talk about the future. And this represents the last of our talks. Hopefully we're saving the best for the last. This is really, I guess, more of a visioning talk rather than talking about practical aspects. But it's also trying to look at issues relating to metabolomics, where it needs to go. You've seen plots for the number of papers for NMR-based metabolomics, plots for the number of papers for mass spec-based metabolomics. But if you just put in the word metabolomics, metabonomics, or metabolome, or metabolome, you can see that, in fact, the number of publications is growing quite rapidly. And this year, there'll probably be over 3,500 papers. So this is growing exponentially, or super exponentially. And it's actually growing much faster, at least in terms of rate, than many other omics disciplines. So it's hot. It's really picking up. So even though you're coming in a little late, you're actually probably all pioneers, given that this still has a long ways to grow. But there's an open question, which is, where is metabolomics going? Will it continue to go up? My feeling is yes, but it could also start flattening out, which has already happened with proteomics and genomics. Or it could crash and burn. It could all find that it just isn't working. So this is an issue that's been thought about, I think, by a fair number of people in the area of metabolomics. Many people have been involved in it. And how can we sustain it? How can we ensure that it maybe grows reasonably? Maybe it doesn't have to grow exponentially. But what are the things that are fundamentally limiting metabolomics? Because if it becomes too difficult, too costly, or if the information that we're generating from metabolomics isn't telling us anything useful, then it will start plummeting in both interest and use. So I've looked at this, and many others have, and I think we've identified some common bottlenecks in metabolomics. One is something that relates to the lack of automation. I think all of you have experienced or have tried a metabolomics experiment, and you find that you're spending months in the lab and months on the computer doing a lot of manual entry, or manual loading, or manual sample preparation, or manual data analysis. That's one. I think another central bottleneck is the fact that there's very incomplete metabolome coverage. Whereas in genomics and proteomics, we're measuring thousands of named genes and named proteins. Generally, people are really, really happy if they can identify 40, 50, maybe 100 metabolites. And we know that the metabolome is much, much larger. We know that it's probably on the order of anywhere from 600,000 to 2 million compounds, of which 95% we don't know of. There's also a challenge in metabolomics that it uses a lot of equipment that is expensive and hard to use. It's not trivial to run an NMR or repair an NMR. It's not trivial to run a mass spec or run a mass spec or repair it. Another issue with metabolomics is something I've been harping on a lot for this course, is the lack of quantification. When you can get absolute values, real concentration values, it means that it doesn't matter what platform you're measuring on. It doesn't matter which place you're measuring in, which country you're from, what instruments you used. Those things are consistent and reliable and reproducible, and that makes the field reproducible. And in fact, that's been a bottleneck that's largely killed proteomics over the last 20 years. I think another issue has been the ability or inability to translate findings into the clinic. And when I say clinic, it could just be about anything. So it's into the field, into veterinary practice, into food industry, whatever. We've been stymied with that. And in fact, we've been stymied that in many fields of omics science. And then the last one is just trying to make metabolomics matter, particularly to drug companies, because they were the ones that launched it in the late 1990s and who've basically walked away from it. And if we could re-engage them about the importance of metabolomics, then in fact we might also see a fair bit more general interest in metabolomics across the field in all life science disciplines. So I'm going to talk about each of these things. I'll explore them a little bit, elaborate on them, and talk about both the challenges and then some of the solutions for them. So first one I'll talk about is automated metabolomics. And these are some important trends. So we're going to look at where people are addressing them. So automation, expanding metabolome coverage, making metabolomics devices portable, getting into quantitation, translating metabolomics, and moving metabolomics back into drug development and discovery. So in the area of automation, in fact, it's already happening. I've mentioned this a few times. Brooker is actively doing this. LipoScience was actively doing this before it was bought up by LabCorp. But these are the tools. This is an instrument for automatic sample collection and handling, things like the wine and juice screeners that they've developed, the software that they've developed to allow you to rapidly load, rapidly quantify, rapidly identify components in wine, juice, and beer, or honey. There's also automated tools like Biocrities, which automates a lot of aspects of mass spectral-based metabolomics. You can routinely measure 180 different compounds with their kits, and you can process samples in as little as 100 samples in 36 hours, all automatically. We've talked as well about automation with NMR using freeware like Basil or Batman, and how it works. And we've gone into some of the details for that, but that is a clear trend. And with the concept of kit-based NMR, also suggests that that's going to happen. So this should make it much more accessible to people. I've heard a few people as well also ask about, I'd like to do some GCMS, and how can I do that? So there's automation that's happening with GCMS as well. We've been working on a web-based tool, unlike Basil. It's called GC AutoFit. And it's a little more elaborate, but it requires that you have spectra both of the sample of interest, a blank to deal with the background, and a set of alkane standards, which allow you to calibrate your attention indices. And it does the alignment that we've talked about. It does the peak picking. It does peak integration. It does concentration calculation, and accepts a bunch of different files. It's actually quite a bit faster than Basil, and in many cases it identifies more compounds than Basil. But remember, we're looking at GCMS, so it's a different platform. Anyways, we've worked it out so that it works quite nicely for serum and for urine, for saliva, for cerebral spinal fluid. There are protocols that are accompanying with it that you can get from the website. But it's pretty easy to use, and it too will actually be converted into a kit over the next month or so, so that would come with all the reagents and protocols for people to run it. So those are examples, whether it's commercial automation or whether it's freeware automation that's being produced by different labs. And again, we're not the only ones doing it, but it's just an example of where things are going, trying to make compound identification, compound deconvolution faster, easier, cheaper, and a shift largely towards targeted metabolomics as opposed to completely untargeted metabolomics. Now, another area where there's a lot of work going on is the idea of expanding metabolome coverage. We've talked about some of the limitations with metabolomics and different platforms, lack of sensitivity in NMR, but at least as we start moving up into more sensitive platforms like LCMS methods, we're seeing 5 and 10,000 peaks, of which we can only identify 5%. And that term, the unknown unknowns, the dark matter, that's a real problem. And it's really limiting where LCMS can go and where metabolomics can go. So one question is, what are all these unknown unknowns? I would argue, and I think there's really good evidence for this, that these are actually the metabolites of metabolites. So taking the compounds that we have ingested or consumed or naturally produced by our bodies, and our enzymes, whether they're in our livers or in our gut or other enzymes around the body, transform them again, simply because they don't fully recognize them, or simply because they make mistakes, or simply because they think they're foreign and they need to be transformed so that we can get rid of them. And so there's some examples of metabolism, if you want, of starting metabolites where things get broken down into a whole range of different components. One way of thinking about the unknowns is to think of it in this way, which is we largely know what components are in our foods. There's about 30,000 compounds that are compiled in databases like FoodDB. We largely know what the drugs are. They're in databases like DrugBank. We largely know what are endogenous metabolomes. We also know many of the contaminants and pollutants that are out there. So those compose or represent lists of known compounds. And my best guess is that somewhere around about 120,000 known compounds that at some point or could be in any one of us or perhaps are in all of us. And what happens is that those compounds, whether they're in our body or in our gut, which is the blue stuff there representing bacteria, or just simply by the fact that we're taking these things because they're sitting out in the environment through sunlight or ozone ocean or whatever else, are transformed into this green goo, which represents mostly the unknown compounds. So if we know what's there, and we know or have an idea what actually goes on inside us and around us, maybe we'd have a better idea about the unknowns. So in some cases, we can actually predict what's going to happen through metabolic action, biotransformations, and chemotransformations. So one possibility is to take all of the known metabolites, all of the known drugs, all of the known pollutants and contaminants and industrial chemicals, all of the new known food constituents and food additives, of which represents 100,000+ molecules, and start preparing or synthesizing their metabolites. So this could be a really exciting organic synthetic project for a lot of people. It could be where you simply add material to an artificial gut or an artificial liver and start processing them. Anyways, if you think that the total number of compounds that are represented in that green goo is somewhere between a couple million to 5 million compounds, and if the preparation and isolation of each of those is going to cost about $1,000, it comes to a total cost of $5 billion. 5 times more than the Human Genome Project. So we've pitched the idea to the US government, and they turned it down real fast. So the alternative was to do this computationally. And in that way, it actually would be a lot cheaper. It means, of course, none of the compounds are made, but they're virtually made. So the question is, how would you do that? How would you do it computationally? So if we take the set of known compounds, which in this case is a little dated here, but it should be, let's say, on the order of about 100,000 known compounds. What you would do is go through a process of predicting the biotransformations, the chemotransformations, predicting what's going on in the gut, in the liver, with other enzymes through phase 1 and phase 2 metabolism. So from there, you go from 100,000 compounds to, let's just, for the sake of argument, say 5 million compounds that are feasible. So from that, you have a set of structures, 5 million structures. Then you go through the next step, which is to predict their observables. Because we can't observe structures, but we can observe spectra. So you can start predicting their GCMS spectra, their MS spectra, their NMR spectra. So we now have 15 million spectra, one proton NMR spectrum, one MS spectrum, one EI-MS spectrum for everything. And maybe we predict the retention indices and maybe a few other parameters, IR. But these are the observables. So now we have a link between the observables, the spectra, and the structures, and also the enzymatic processes that lead them or led to there, as well as their origins, because we started with a set of compounds. And then we can do the last step, which is simply matching the observed spectra to the predicted spectra to identify all those unknowns. So what we're working on, and others are also, is a tool called Biotransformer. So in our bodies, there are 2 types of processes called phase 1 and phase 2 metabolism to get rid of xenobiotics or drugs or pollutants. Those are mostly done by the liver. There are lots of things that will take compounds that we're exposed to or outer environment will transform them. So the microbes in the environment will transform things. Our gut microflora will also change things quite dramatically. And then there are promiscuous enzymes, which don't have perfect substrate specificity, and they'll take certain kinds of compounds. Alcohol dehydrogenase is a good example, which will convert alcohols to aldehydes, but a whole bunch of them, not just ethanol. What we've cataloged is a little over 600, almost 700 different biotransformations, and have developed both machine-learned rules as well as heuristic rules that identify where the sites of reaction will occur, where the transformations will occur, and that also generate the structures. So this is an example of a biotransformation prediction with the biotransformer. So it starts with a polyphenol called quercetin, which you can find, I think, in onions. And this is what happens. It takes quercetin and it starts running it through its enzyme list, and it starts transforming it, changing some of the hydroxylation patterns, opening different rings, going through 1 step, 2 step, 3 steps, 4, even 5 steps, generating a bunch of metabolites. And what's shown are actually metabolites that have been identified of quercetin. So this is what is predicted, and these are the ones that are known. So it's actually doing a pretty good job in terms of identifying these structures and doing the chemistry or enzymology. The ultimate product of most polyphenols in our bodies is hippuric acid. And that's typically a conjugation of sort of a benzoic or phenolic acid with glycine. So this is just illustrating the types of things that biotransformer is able to do and the number of steps it can do. And what's more is it's not just generating random structures, it's actually giving you pathways. Okay, so you've got 5 million structures. What do you do with them? So one approach is to start predicting their spectra. And we've been spending a fair bit of time over the last 2 or 3 years developing software to predict both electrospray ionization, MS spectra, and more recently, electron impact, MS spectra. And so the tool, which is called CFMID, it's available on a website, it's also downloadable, is capable. It's, as far as we know, it's the only freeware out there that allows you to accurately predict MS spectra and EIMS spectra. Now, it's not perfect, but there's also software that we've talked about as well that's available for predicting NMR chemical shifts from their structures. And this is an example I've shown before, but take a very complicated structure on the right, and you can see the observed spectrum and compare it to the simulated one. So right now they're mostly commercial predictors, but the fact that they are available and the fact that they're this good also suggests that it should be possible to predict the proton NMR spectra and probably the C13 spectra and the TOXI and COSY spectra for all 5 million compounds. So what's happening, or at least over the next little while, it looks like A, there are going to be tools that allow people to confidently and consistently predict metabolites of metabolites, and B, that there's a real trend in being able to predict spectra, both MS, EIMS, MS, and NMR spectra quite accurately. And so this will create, I think, virtual databases of virtual compounds and virtual spectra that should help tremendously in identifying those unknown unknowns. Already people have actually taken not the structures, but the predicted masses of these biotransformation products. And they can get up to 90% of the compounds, the features in MS spectra, LCMS, tentatively identified based on the mass matching. So that's, again, very encouraging. That suggests that most of those unknown unknowns are really just metabolites of metabolites. How can we make metabolomics more portable? Right now, metabolomics is very elitist in the sense that in order to do it, you have to have multi-million-dollar facilities. There's lots of investment. Some of the best spectroscopy facilities in the world do metabolomics. But it means that it sort of makes it somewhat isolated in the sense that it's not there in every lab, it's not there, that it's not as accessible, physicians can't use it, vets can't use it. And that's a problem. One of the things that's starting to happen is that we're seeing, whether it's on your iPhone or your Fitbit or some of these new developments called sensors, which are essentially trying to move the Star Trek tricorder out from Star Trek and into reality, where people can actually track their health. Now, some of these things are mostly measuring your heart rate or your walking rate or things that are pretty trivial. But could we do the same for metabolomics, where in fact a watch or a smartphone or some kind of patch actually does metabolomics? So we've been starting on an initiative, and others are also, which are the idea of democratizing metabolomics. So you can take a 900 megahertz or a gigahertz NMR, and those are $9 to $10 million. And it's also possible to shrink things down. And what's shown is actually a handheld metabolomic device that's been around since about 2005. It does a whole bunch of portable clinical assays. And it's cheap, and the costs per test are cheap. It's not great in the sense of it's not measuring all kinds of different metabolites. It measures standard things like glucose or carbon dioxide and oxygen. And every test requires a different pack that you have to upload. And so it's not the most popular thing, but it is suggesting that you could democratize and make metabolomic tests a little more portable. Some of you might have heard of the X Prize. There are X Prizes that are offered for, pedaling across the Atlantic on a man-made or human-powered flight system, or X Prizes to go out into space for more than a few hours. And one of the Qualcomm X Prizes is to actually make a tricorder. And in fact, the first prizes have been awarded. And again, it's mostly a tricorder that's based on measuring heart rate and pulse, temperature, and things like that. But there were some of the entries that were measuring compounds. And some of those entries were starting to use things like miniaturization and microfluidics and nanotech, capillary electrophoresis on a chip, HPLC on a chip, gas chromatography on a chip. Those are all possible. They're being done. Efforts in several labs around the world show that these are consistent. Some of them are even commercialized. There's actually quite a rapid advance in the development of volatile metabolite detection, electronic noses, which are, again, handheld instruments that allow you to detect different volatiles and measure their abundance. They're not perfect. They represent patterns rather than quantitation or exact identification in some cases, but they're trending towards being more quantitative and more precise. In our own lab, we've been thinking about trying to move away from mass spec and NMR to the idea of using antibodies and paraplasmic binding proteins. Paraplasmic binding proteins are essentially antibodies that microbes produce to bind to metabolites. They're very metabolite specific. They sit in the paraplasm, and they move about 40 different types of metabolites from outside the cell to inside the cell. One of the concepts here is to actually attach gold nanoparticles to the metabolites and then use competitive binding assays with paraplasmic binding proteins or metabolite-specific antibodies to permit detection. And when you've got nanoparticles, it actually allows you to do some really simple detection. You can use things like impedance or electrical detection. And those are very small and very simple to prepare, very sensitive. You can use things like surface plasmon resonance as well, or surface-enhanced Raman spectroscopy. And these are things that are maybe not quite the size of a smartphone, but they're maybe the size of a small toaster. And they're certainly something that you can afford readily. We already have a working prototype, one that uses impedance. It's got Bluetooth connections, and it uses specially designed chips, specially designed electronic systems for detecting impedance, and it's able to detect about 3 or 4 different metabolites. There are other applications, other areas where people are moving towards, particularly with lateral flow assays for metabolite detection. And to multiplex those lateral flow assays, again, should give people the possibility of detecting metabolites with just basically strips of paper and at a cost of pennies per sample. Another area where metabolomics is going and has to go is the trend toward quantification. So about 2 years ago, we did a study and found that more than 90% of published metabolomic studies are semi-quantitative. Either they report relative peak areas or intensities or no quantitation at all. And less than 10% of metabolomic studies that were published use absolute quantification. Now the trend is improving. It's probably now 80-20, but it's still a long ways from where it needs to be. And if any field is going to advance, if anything is going to be used outside of the lab, it basically has to be quantitative. And this was actually a cover from Trends in Biotechnology from, I think, 2 years ago. And the title cover is, "There's a specter haunting metabolomics, the specter of quantification." So this was identified, not just by me, but certainly by many others in this review article. So quantification is happening. It's happening again, with Brooker's systems for automated NMR, with Genomics and their system for spectral deconvolution, with Biocrities for their tools for automated mass spec. There's academic efforts for quantitation. I've talked about Basel and Batman. We've just mentioned GC AutoFit. So these are happening. They're available. Some of them are commercial, some of them are free. The point is there's nothing really that prevents you from using these things, and you should be using them. I've highlighted, I guess, a contrast between what metabolomics and proteomics and transcriptomics can do in terms of their quantification records. So in transcript analysis, there is no absolute quantification. With RNA-seq, there's maybe a trend towards that. In proteomics, quantitative proteomics has just started to get discovered. And right now, the best and highest number of proteins that we've seen fully quantified and identified in blood or serum was about 73, in CSF about 130, and in urine about 63. If we look at metabolomics in terms of what's been able to identify and quantify, using either one platform or multiple platforms, we can see that, in fact, metabolomics wins. And this is an important thing, that saying that, in fact, metabolomics people are getting the picture. They are working to quantify. They're, at least for now, ahead of the game in terms of proteomics and transcriptomics. But I think a lot more can be done, because these only represent a tiny fraction of the total metabolome. Fifth thing that I wanted to talk about is the idea of moving metabolomics from the lab to the clinic. Now, again, I'm focusing more on medical applications, but it's still an issue of how to get things out from the lab and translate it into general use. So in the last 45 years, there's been more than 700,000 biomarker studies published that show up in PubMed. In the same 45-year period, there's been fewer than 250 biomarkers that have been approved for use. Right now, there are no proteomics biomarkers that are approved. There are protein biomarkers that are used or approved that use ELISA, but not ones that are based on MS. And there are 5 biomarker tests that use transcriptomics. So again, that's looking pretty grim. But there's an interesting statistic, which we talked about before, that actually metabolomics has already been in the clinic, although we just didn't know it, and that most of you probably have had a metabolomic test, and again, you didn't know it. And so this is newborn screening. So in some regards, metabolomics has already made that transition, and it made that transition into the clinic long before proteomics, ELISA, and genomics did, because this has been around for more than 25 years in most of the developed world. So in one regard, metabolomics has a leg up. In another regard, metabolomics also has a leg up on the other techniques. And this is, again, something that I pointed out last, I guess, a few lectures ago, that in terms of the number of approved tests that these different omics fields offer, metabolomics/clinical chemistry actually has more. It has more than genomics, it has more than protein tests, it has more than transcriptomics, and certainly more than proteomics. So in this regard, metabolomics has been successful, but really what's happened is no one has told the funders or the people up at the top that in fact that it's succeeding despite the relatively minimal investment that it gets. What's more, metabolomics also seems to exhibit a much better performance, and metabolite profiles generally seem to exhibit a much better performance than many protein, genomic, and transcriptomic tests do. So we saw some of these examples before about how metabolomics does in terms of predicting disease. And obviously, if you can predict disease, it's often something you can do to prevent it. And we talked about how you can use it to predict preeclampsia in expectant mothers, how it can be used to predict congenital heart defects at very early stages in pregnancy, or predict the presence of chromosomal abnormalities in fetuses, or how it can be used to predict which individuals will develop cancer cachexia months before it actually occurs. And whenever you can do that, whenever you can predict things 3 months, 5 years, 15 years in advance, that makes a big difference because there are things that you can do to prevent that. You can also use metabolomics to diagnose diseases and diagnose those that are really hard to characterize. And again, these are examples of ROC curves, which are all above 90%, for diagnosing difficult to diagnose diseases like kidney rejection or systolic and diastolic heart failure or chronic fatigue syndrome, or for screening purposes in the case of polyps. And I've already mentioned, in fact, that the polyp test has now moved from sort of the lab to the clinic as a 3-metabolite test. So metabolomics actually is moving from the lab into the clinic, but the message isn't getting out. And that's unfortunate. And I think this is something that anyone who's involved in metabolomics needs to worry about, is if the message of its successes aren't getting out, then it's usually going to be the one that gets cut. And somehow some of the other fields that in fact are often less successful, transcriptomics, proteomics, even genomics, are somehow getting their message of success out, and in fact continue to grow at a substantially faster pace than metabolomics. So, not sure whether it's by deed or by description, but somehow I think the message needs to be delivered that in fact metabolomics is succeeding in the translational process, but certainly we can do more. Another area where I think metabolomics trends that are happening moving forward are getting metabolomics back into the field of drug development and discovery. So this is a typical drug development pipeline where there's a discovery phase that take 3 to 4 years, a phase 1 trial, a phase 2, a phase 3 trial, and then final drug approval. The whole process can take on the order of 10 to 15 years and can cost over $1 billion for each drug. Now, there are different roles that different fields will play. So in the discovery phase, there's a lot of medicinal chemistry. Also in the discovery phase, there's often a fair bit of genetics and genomics that used to help identify targets and sometimes in identifying patients who are susceptible or best suited for that. Often proteomics complements those discovery early phase works. But what people don't realize actually is that metabolomics can play a role from discovery all the way through the trials and finally approval. And this is illustrated here. So discovery can be part of identifying markers of disease and therefore identifying targets. Discovery can be also part of the use for rapid toxicology screening, looking for preclinical efficacy markers, and to also help with lead prioritization. Once you move to the safety biomarker screens and mechanisms, then you can start switching into phase 1 and phase 2 and phase 3. And again, metabolomics allows you to identify the safety biomarkers and the efficacy markers all the way through. So here's some examples where you could take a bunch of patients who've volunteered to take the drugs in phase 2 and 3, and with the explicit requirement that they cannot drink. So if it's just dependent on someone saying yes and no, of course they'd all say didn't drink. But this is what metabolomics allows you to do. You can also use metabolomics to identify the fast and slow metabolizers. And in many cases, that's not done in the phase 2 and phase 3 trials. And if you end up taking a patient who's an extraordinarily fast, they call them ultra-fast, or an extraordinarily low, an ultra-slow metabolizer, those people can die from these drug treatments. And if you have deaths during a phase 2 or phase 3 trial, that's the end of the drug, and that's millions or billions of dollars lost. So by looking at whether the drug metabolites are persistent or rapidly disappearing, it allows you to determine whether these patients are suitable or appropriate, or whether the dosing should be changed. So these are applications that can be done or can be used in drug discovery and development. Unfortunately, they're not. Now, in traditional drug discovery, it's a very long-shot process. Right now, most drug discovery efforts are based on large-scale genome-wide association studies or proteomic-wide association studies, where they look over large cohorts and try and identify particular genes, SNPs, proteins, whatever, that are potentially druggable, targetable. These generally lead to a success in about 1 in 5, even though each of these studies costs between $2 and $10 million each and takes between 2 and 4 years. Among the targets that they identify, only a half are potentially druggable in the sense that they're actually targets that you can clone or generate, or that are stable enough to study in any degree, or for which you could develop an assay for. And of those, not all of them turn out to be sufficiently stable or sufficiently purifiable for any kind of assay. So there's maybe about a 50% success rate in each of those steps. When you finally do have a drug target that's stable, easy to handle, that's suitable for an assay, then you can start doing drug screening. And so we have roboticized, high-throughput screening systems that allow you to identify ligands that bind or inhibit or activate the proteins of interest. But not all of those screening assays work. On average, about 20% are successful, and the screening assays can run for years. Once you have a lead, one that seems to behave well, seems to work, at least in the test tube, you can go to the next phase of drug development, and that's where you only have a 1 in 500 chance of success. And the statistics on that are out. And instead of taking 1 year, 5 years, it takes about 15 years and costs about $1 billion. And then of the drugs that are approved through phase 3, about half are either taken off the market because they don't sell, or prove ineffective, or are too toxic. So if you multiply the probabilities of 1 in 5, 1 in 2, 1 in 2, 1 in 5, 1 in 500, 1 in 2, you get to essentially a 0.001% success rate in an average of 20 years, and well more than $1 billion to get drugs out. This is the main reason why the drug development pipelines have dried up for every major pharmaceutical company. It's not working. On the other hand, you could think about trying to do metabolite-based drug discovery. Based on our work and work of many other people, where we do metabolite-wide association studies or assess trends and biomarkers in cohorts and groups, it's often possible to identify not only metabolites, but also the corresponding genes and enzymes. And I've talked about this for the trimethylamine oxide story, the diabetes story, the oncometabolite stories, and on. And in many cases, those are relatively inexpensive studies that allowed people to do a lot of the analyses in a relatively short time using tools like Metabolomics, where you could figure out not only the compounds, but also the pathways that they were involved in. And then using simply a software that I've showed you guys, identifying what are the potential targets, and in some cases, what are the potential drug leads that you could use. And in many cases, whether it's things like olive oil to prevent heart disease, or changing gut microflora through probiotics, or targeting specific oncometabolites with certain inhibitors, most of which are in the food or in natural products, you can actually find either existing drugs, and the example for cancer has been metformin, and also the statin drugs, which act on many of the targets that have been identified through metabolomic studies. So either existing drugs being repurposed or existing natural products being used in the proper way, it's possible actually to find products that work. And then you can also use metabolomics to monitor how well these things are doing. If you calculate those numbers, those times, those probabilities, this argues that you might be getting around a 15% success rate, and that you could get potentially usable drugs or drug leads within 1 to 2 years at a cost of $250,000. And this, as I said, is really what had happened with the TMA story and the link to olive oil and 3,3-dimethylbutanol. So it is possible to have something like metabolite-based drug discovery, and it's something that I think the drug companies need to think about, given the fact that their current model is so unsuccessful. So just to wrap up, I think the future of metabolomics is bright. I think there's lots of opportunity. We've certainly talked a lot about NMR-based metabolomics, but this has been a presentation about metabolomics in general. I think we're seeing how, in particular, NMR is highly suited for automated metabolomics. I think we can see how NMR will be playing a key role in expanding the level of metabolite coverage. I think we can see how NMR is playing a real key role in the quantification of metabolites. And it's also encouraging people to come up with more portable approaches to doing metabolomics, particularly with the development of small benchtop NMR instruments. I think metabolomics can also, or needs to be, moving from the lab to the clinic and back into drug development. And particularly in the area of drug development, I can see where NMR-based metabolomics could play a very critical role. So whether it's NMR or mass spec or GCMS, I think all of these technologies, along with some of the newer techniques that I've mentioned to help democratize or miniaturize metabolomics, I think all of these will be playing a key role in the future. And of course, it's not just the equipment, it's the people. And I think this is what we've been trying to do for the last 3 days is to, I guess, get bright people trained up into the area of metabolomics so they can come up through the ranks, come up with even better ideas than some of the ones that have been mentioned here, and improve the whole field.